So I think I would like to move on to the second speaker of the day, who's Mar Albà. So it's a pleasure to introduce Mar, who has been working on understanding the molecular evolution of microproteins and gene birth in various organisms. Mar obtained her PhD at the University of Barcelona in Spain, followed by a master's in bioinformatics and molecular modeling at the University of London. She then did her postdoctoral research with John Hancock at the Medical Research Council and Paul Kellam at University College in London. And currently, Mar is a professor at the Hospital del Mar Research Institute in Barcelona, Spain, and the Catalan Institute for Research and Advanced Studies, where she leads the evolutionary genomics group. Mar's research on microproteins covers diverse topics in organisms, including evolution and de novo birth of microprotein encoding genes in yeast, and the diversity and function of microproteins in cancer. The title of Mar's seminar today will be How Microproteins Contribute to Evolutionary Innovation and Cancer. Again, please post the questions you have in the Q&A window or ask them after Mar's presentation. So Mar, you can take over now. Mar, you're still on mute. Maybe Lauren has to. Yep. Perfect.
Here we are. I was saying that thank you very much for this invitation to have the opportunity to talk about our research. And I will start presenting some ideas of what brought us to microproteins. And that's a while ago, nearly 20 years ago, when we started to be interested in a type of proteins that are called orphan proteins, or orphan genes. And they appeared when the first genomes were being sequenced. And here I'm showing a very old plot where the researchers were performing comparisons of the genes that they could find in two yeast species and in C. elegans. And they noticed that there were a number of proteins, quite a high percentage here, 20% of the proteins that were Saccharomyces cerevisiae specific, meaning that they could find no homologs in other species. And that was quite mysterious. So what was the origin of these proteins? It was quite unexpected when these comparisons started to be made. And we got interested in this, and we decided after a few years, when we already had a number of vertebrate genomes and metazoan genomes, we performed also some of these comparisons, looking for proteins that were in humans and also in mouse that were conserved in the two species. So we could be sure that these proteins really existed. And then we compared them to the proteins or the genes in the other genomes, and we found again that there were these orphan genes that were specific of human and mouse in this case, and they corresponded to a small protein. So we were here already in the field of microproteins, and they were also rapidly evolving. They evolve faster than other more conserved proteins. So at the time we didn't know if this fast evolution was the reason why we couldn't see the homologs in the other species, or there were other reasons for that, and these proteins were actually evolutionary novel, or here we started to hypothesize, to think about this possibility that they had been generated de novo. And I'm going to explain what this means and how it's related to the microprotein field. So de novo generated genes arise when a fragment in the genome, region in the genome, starts to be transcribed, and then it can also be translated, giving rise to a completely new protein. And we started to find the first evidence of this process in studying humans and primates, these primate orphan genes, where we could perform these comparisons, because then at the time, it was 2009, we already had some well-sequenced genomes of different primates, so these were quite closely related, another mammalian species. And we could see that some of these proteins would be clearly detectable and even conserved between human and macaque. But when we look at the genomic regions in the other species, they would not be able to translate these proteins. So you could align the genomic sequences, but then you could see that there was no protein there. And you could go farther and farther. Sorry, you could go farther back. And see no evidence of homologs in other species. So these were really novel proteins. And here, yes, we could say that they had originated de novo from these non-coding genomic regions. So these were very exciting times. At the same time, other groups were detecting these kind of proteins in Drosophila or in yeast. And it was a novel mechanism that we were uncovering for the formation of new genes. So this de novo gene formation implied that we would have proteins with completely new sequences derived directly from non-coding parts of the genome. This concept is still relatively new. So this means the sequences really, they come from parts that are initially non-coding, and then they become translated, and then they start to be produced in the cell. And some of these proteins, they continue to evolve and acquire new functions. And this is, of course, very different from gene duplication, for example, where new genes will be formed. Initially, they will be identical to the other copy, and then over time, they will also tend to diverge, but always maintaining sequence similarity and structural properties that are similar to the ancestral gene. Whereas here in de novo gene formation, we have a very radical process of generation of new proteins. But how many de novo genes are there? Because these proteins are small, and automatic methods to annotate proteins or in genomes or even in transcriptomes are based on the size of the open reading frame. We knew from the beginning that we could be missing many of them. So the annotated part, the annotated in the databases part was probably much smaller than the non-annotated part. So probably there were many more of these de novo small proteins that we couldn't see, and that they could be in transcripts that were annotated as non-coding. Luckily, as Sebastian explained very well, there was this method called RiboSeq, ribosome profiling, that is very, very sensitive and can detect very small open reading frames that are being translated. So using this methodology, we could get to all these small open reading frames. And these early works from Nick and Golia, who developed this technique, already show that there was this translation patterns using the read from this technique, indicating translation of ORFs or small open reading frames in long non-coding RNAs. So indeed, this technique allows us to unleash all these small open reading frames being translated. We continue to investigate this over time, and with more species being sequenced, with now some ribosome profiling data from humans, also some proteogenomics data, and studying the properties of these de novo genes that were found in humans or only in other related primates. And what we could see when we plot them together with open reading frames from introns or from or small coding sequences that are annotated, we could see that the size of these new de novo genes, de novo proteins, is similar to what you can expect by chance in the genome or in intronic regions here as a proxy, but that they, from their birth, they tend to have already certain codon usage bias, meaning that the codons that they have are more like the ones in the proteins, that the coding sequences that encode most of the proteins we know. And this is because of biases in the sequences that are being transcribed. They are more GC rich, and they tend to be from the very beginning to have higher frequencies of the codons that are then more frequent, that we are going to see more often in canonical protein coding sequences. Ribosome profiling data continue to accumulate, and we continue to be very interested in this question of trying to understand how these de novo genes originated, and using this data was ideal to have a very complete image of what was going on. And here in mouse, we could use data from different types of cell lines and from different tissues and combine it and to uncover more than 1,000 small open reading frames that were translated. And then we could, in long non-coding RNA, so this would correspond to putative de novo genes, but then we look at the conservation using data from other related species, from rat, from other mammals, from other vertebrate species. And then we got new clues about how many of these small ORFs in the long non-coding RNAs were conserved in other species, whatever species, so they were not mouse specific. And then using SNPs, I'm not showing the details here, but we could use data on the variability of these sequences in wild populations of mice, also to get hints about the strength of natural selection, if there was purifying selection, which is then you can link it to functionality in different subsets of these more than 1,000 small open reading frames. And we could see a group where we could see some conservation and some selection. So this group probably contains conserved functional proteins. The only thing is that they are small, so they had not been annotated, and we could only see them here for the first time. But then there were many that were not conserved. So that were in transcripts that appear to be really mouse specific. A few had, well, a small group, let's say, had some signatures of purifying selection. So this could be mouse-specific functional proteins, or at least some of these proteins in this green part here were probably functional, but very young. And then we could also see a big group where there was detection by ribosome profiling, which, as we have seen, it's a very sensitive technique, but we could see no conservation, no selection. So we have this group of putative, the way we interpret this data here is that they are putative precursors of new genes. So some of these could acquire functions, and this would fit into this schema of how new genes are formed, different stages. So we will have a lot of events that correspond to pervasive transcription, translation, small proteins are being generated, not necessarily functional, but some of them then acquire functions, and then some of them end up being conserved in other species and becoming established. And of course, there must be a lot of turnover as well, these initial stages, because otherwise species will continue to accumulate many more genes than the ones that they are accumulating. So at the end, we see when we analyze lineage-specific genes, we see that there's sometimes, well, there's some of them, like even hundreds of them, but it's not continuous growing of the number of genes that we have. So there's a lot of turnover in these initial stages of species-specific proteins. We also have investigated this question of de novo genes, trying to get full catalogs of de novo genes and microproteins in yeast, and doing these comparisons across different species to get information about the evolution, how well conserved these microproteins are. And in a recent study, we identified more than 200 genes in Saccharomyces cerevisiae that we classified as de novo. So when we look at the genomes, the synthetic regions of the other species, we could see no evidence of similar genes. So again, these genes have appeared from non-coding parts of the genome. Before we did this study, there were few examples known. One is BSC4, a DNA repair protein. But here we are really expanding a lot, from a couple of examples to more than 200. And this represents 5% of the Saccharomyces cerevisiae genes that appear to be very young, classified as young and born by the de novo mechanism. These genomes are very compact, the yeast genomes. So what we discovered is that many of these new events of transcription and translation that make the microproteins happen in antisense direction to other genes. So the two strands are being used in this genome to extend that was not known to make new proteins. Some of these are involved in stress processes, like this duplication here, CAP1, which is a gene that has already been characterized, but then we discovered in this work that there was another antisense transcript containing two open reading frames that had been duplicated together with this other gene. And the two transcripts, sense and antisense, are overexpressed in stress conditions. So probably both participate in the response to stress. And many more examples of genes that we still don't know what they are doing, both in antisense, which made about half of the genes here, or also in intergenic regions like the BSE4 example. Right now, we are moving to even closer evolutionary distances. We are trying to see if these de novo genes can be detected when comparing strains, when comparing different yeast populations around the world. So here, instead of millions of years, we are talking about thousands of years. And we want to see these events of emergence of new proteins, that some of which may be adaptive. And we are comparing, for this, we are using complete genomes of, in a pilot study, we started with 7 strains, and we already see some of these events. And now we are scaling up to have a better picture of how frequent de novo gene birth is at the level of these strains. And now I like to talk a little bit about these microproteins as a source of tumor-specific antigens. Several years ago, as Sebastian also mentioned, there was some research, particularly a paper that really for me was made a change the way I thought about these microproteins and their possible roles in cancer. And was this paper where they observed that many of the majority of the tumor-specific antigens, so this would be the peptides presented by MHC1 receptors, they came from non-coding regions. When you try to match the data, the immunopeptidomics data, with the non-proteins or the non-coding transcripts, many were coming from non-coding transcripts. So that's, of course, very, very exciting, because means that some of these could be targeted for therapies. For example, they could be used to make vaccines that specifically target or stimulate the immune system to destroy the tumors. And so we have done some research in a system which is hepatocellular carcinoma, where there aren't many mutations in these tumors that immunotherapy therapies, that the immunotherapy that is based on the targeting mutated peptides, neoantigens, is not so promising. So here our aim was to quantify the number of tumor-specific antigens that could be generated by these long non-coding RNAs, as a first step towards maybe in the future developing vaccines based on this kind of microproteins. We gathered data from different data sets that are publicly available, RNA data sets, that was to have a view of how these long non-coding RNAs were distributed in different patients, and then also some riboseq data to detect these microproteins with this technique. And we did a catalog, just quantifying the expression of protein coding and long non-coding RNAs, and even novel genes, which would be de novo transcript assemblies using the RNA-seq data in the different data sets. And then also using the riboseq data on this transcriptome to identify ORFs that are being translated. And this also was very nicely explained by Sebastian, where you have the RNA-seq patterns that cover the entire transcript, and then you have other signals that are specific for the open reading frame. And we could see hundreds of these open reading frames that were being with evidence of translation. And then we look at the fraction that was tumor specific. This was very, very important because it's the kind of antigens that could be useful as vaccines because they are only found in the tumors. And so stimulating the immune system to see these antigens could make the immune system to fight more strongly against the tumor, and perhaps even in the future help to cure these tumors. And we observed that the tumor-specific transcripts were actually enriched in long non-coding RNAs compared to protein-coding genes. So they are an important component of this tumor-specific transcriptome, these long non-coding RNAs. This was the first message of this work, the first discovery. And then we predicted peptides, how many peptides they could have that were presented as antigens that could bind to HLA MHC1 receptors. And we also validated some of them. The validations were useful to see that 70% of the predictions that we made here could be seen then in an in vitro assay. So the predictions were relatively good to get an estimate of the contribution of these ORFs in the long non-coding RNAs to generate these tumor-specific antigens. And at the end, we got a view of the contribution of different types of antigens, neoantigens, in these hepatocellular carcinoma cells in the different data sets. We see similar results. So what we could see is the number of epitopes or antigens generated by these ORFs in the long non-coding RNAs was very high, represented about 40% of the total of the neoantigens we could predict that would be presented by the MHC1 molecules. So indeed, it's not the majority, but it's really a very large part, an important part of this immunopeptidome. And then because we had a large cohort of more than 100 patients, we could study also how many would be shared between different patients. This is important because to develop therapeutics means that we could think of strategies that can target not just individual patients, but maybe 10-20% of the patients, and that maybe then combined they could target a large part of the patients. And although the majority of these antigens are patient-specific or present in a few patients, there's also some that are shared by 10, 20, 30% of the patients. So these are particularly interesting. They are tumor-specific. So we think they are valuable as a basis to think of therapeutics that could target these tumors. And with this, I'm going to end. So I want to acknowledge people working on de novo genes. The first part I presented and how we got into the microprotein world. People that work with me over the years, starting with José Castrasana, Macarena, Jorge Ruiz, José Luis Villanueva, Will Blevins, José Carlos Montañés, Chris Papadopoulos, and Kobabara. And also the work I showed on cancer, the long non-coding RNA ORFs in cancer. I've shown work from Marta Camarena, Julia Pereira, Patrick Teunissen, Uri Fortes, and Pablo Sarobe. The last three researchers are working at the University of Navarra. And I also, of course, thank my funding sources. I'm ready if there's any question from the audience. Thank you.
Great. Thank you so much, Mar, for a fantastic overview of the evolution. And I'm sure there'll be lots of follow-up questions. Maybe in the interest of time, I'll ask people to post their questions in the Q&A window, and you can specifically address them to Mar. And I see people have already had some questions answered, so that we can move on to the panel discussion. And then Mar can also answer some of the questions on the evolution. For the panel discussion, we have 4 experts in their field, and I'm going to post some of their recent papers that are relevant into the chat window and also their website, so you can read up more about their research, because we won't have enough time to cover that in the remaining half an hour. But I want to give you a brief introduction of who we have today and then ask a few specific questions to our panel members. So we have Jonathan Mudge, who's joining us from the EMBL European Bioinformatics Institute in Cambridge, UK. His area of expertise is gene and microprotein annotations, database curations, and standardizations. And we've seen his name on some of the slides that Sebastian already presented. Thomas Martinez is joining us from UC Irvine in California. Thomas's expertise is in advanced methods for microprotein identification and detection, including mass spec and ORF finding, and the function of microproteins in cancer and metabolism. John Prenzner is a pediatric neuro-oncologist from the University of Michigan Medical School. John's expertise is in microprotein essentiality, their roles in cancer, and their clinical implications. And finally, we have Lee Zhao joining us from Rockefeller University in New York City. Lee's expertise in ORF detection, de novo gene birth, and adaptive evolution has been applied to Drosophila, and she recently had a beautiful paper on riboseq analysis of many previously unknown genes in Drosophila, or microproteins in Drosophila. Feel free to ask questions in the Q&A window to these experts if you have any. And I would like to start with a question to John Mudge. Many people, I'm sure, are looking for databases of human microproteins or ORFs. So how do they start? What are the currently available databases, and what are we expecting in terms of changes to those databases? And Sebastian already touched on this, but from a practical point of view, where would you go, and what databases can you download to get started?
Sure. So thank you for having me here today, and I'd like to say how much I enjoyed the presentations as well. So as you mentioned, I work on the GenCode Ensemble project, so I'll start with what we have and what we've been supporting. So if you want to talk about what is canonical to say proteins that we agree are proteins, and we could be what I call a reference annotation ecosystem, also with UniProt and RefSeq gene annotation. We have about 200 canonical known microproteins, and these tend to be the things that have been published before, going back many years. And we've also supplemented that in recent years with microproteins, which were really obvious to identify based on their evolutionary signature. So for that reason, we're quite sure they're microproteins. So you have about 200 of those, but then I suppose you're also asking about the rest, so the vast bulk of these potential microproteins that have not been annotated yet. So I'd like to say we're not skeptical about these at all. It's just that we have a really important gatekeeper role in reference annotation, because we're used for example, all clinical science and things like that. So Sebastian already mentioned the list of 7,264 riboseq ORFs. So these are supported by GenCode as an annotation file. You can take it from our websites and UniProt and HGNC as well. So these are not specifically micro, by the way. We're not just interested in micro. It just so happens that the vast majority of riboseq ORFs that are not annotated are under 100 amino acids, so like over 95% of them. So we present that file in a way that's agnostic. So we don't say which are proteins and which are not proteins. We simply say, here's 7,000 riboseq ORFs, and people can go out and have fun with them. There are also other catalogs and publications that people have made as well, things like OpenProt and things like that, which can be larger, sorfs.org and things like that. And I think there's a lot of fascinating questions that then come onto this, is how do we get from saying that these are potential microproteins to actual things that we're certain microproteins, which, as I say, is the point at which we want to actually annotate them. And, I think going back to Mar's talk, we also have this fascinating question of an ORF is translated, but the protein is specifically found in cancer. And then, do we want to call that a protein coding gene, or do we want to call that something else? So I would say the field is really rapidly evolving, and we're doing our best to keep up. I'm happy to take any more questions in the Q&A. slide as
Thank
well.
you. Thank you. That was fantastic. That's a great overview. John, maybe if you can find the link to the 7,000 that Sebastian mentioned and posted in the chat, that would be fantastic.
Sure.
And then this will also be recorded if people want to listen to those resources again. So my second question goes to Thomas Martinez. Thomas has done wonderful work on methodology of RiboSeq and ORF detection. And my question is, what are the remaining main technical barriers for microprotein discovery, and are these likely to be resolved?
Yeah, that's a great question, and thank you for having me here today. So Sebastian and Mar both touched on this earlier, but you know, the primary methods we use to detect microproteins are ribosome profiling and mass spec. The RiboSeq, starting with that, I feel like it's quite worked out. You know, there have been improvements since Nick Ngolia first introduced the protocol to the world back in 2009. You know, improvements with regards to having lower inputs, so starting with less RNA in order to generate your libraries for sequencing. In fact, protocols now have even gone down to the single cell level. But for the most part, my understanding, you know, with these newer protocols, and I haven't actually tried them myself yet, so please, anybody else correct me, is that in order to get to the lower inputs, there's some sacrifices you make on the resolution of the data in terms of that periodicity. And so I don't think it's quite there to the level of you can use single cell data to actually identify novel ORFs as coding, but you could still get good coverage. So it's one of those things where I think you can combine it with bulk data and then kind of get down to single cell resolution in terms of translation, looking at the expression at the translation level of individual microproteins. So that's kind of one barrier that's starting to be pushed. But for the most part, RiboSeq works pretty well, but it has its caveats in that it's only looking at translation. I fully agree with Sebastian. I think a lot of the folks here in that you don't want to, you know, lose mass spec. Mass spec is still that, you know, that last level of detection where you're actually seeing the peptide itself from a microprotein. And there, I think there are, you know, physical limits that we're hitting, but there's advancements that are being worked on. So for example, in a paper we published last year, we collaborated with folks at the University of Washington and Novo Nordisk's proteomics group to apply so-called data-independent acquisition strategies of the mass spec. You know, the fast version to explain this that I like to say is that DIA versus normal DDA, data-dependent acquisition mass spec, kind of lets you have your cake and eat it too. Instead of taking only the most abundant peptides to generate MS2 spectra through a second fragmentation step, you take windows, and therefore you're able to collect all of the peptides, or at least almost all the peptides, and take them forward to fragmentation to actually make MS2 spectra for them. And of course, if you don't have MS2 spectra for your microprotein because it's low abundance and it's, you know, crowded out by the larger peptides, you're not going to be able to match it up to a peptide and detect it. So that's already, you know, coming out now, and it's really been something that's emerged because of the improvements in the technology itself of the mass specs and how much faster they're able to cycle and how much more accurate they're getting. There are still limits with what you can do. You can't amplify proteins, so you can't go quite to the level, get to the level of sensitivity of sequencing. But I do think we'll see more improvements there in the future. So, you know, and then that'll help us answer the big question of whether or not we're missing them because of the method itself, because mass spec is difficult, or because there really is a gap between what's translated and what's actually stably long-lived protein that we can detect by mass spec at all.
That's right. Thomas, have people tried to make synthetic peptide libraries to help identify the native ones?
Absolutely. And that's a very important step to really validate. I would say if you're trying to prove at a mass spec level your individual peptide, it's important to have a, you know, heavy isotope labeled standard peptide that you can see on the chromatogram is the same retention time and will give a similar MS2 fingerprint. And actually, one more thing is that you know, everybody talks about AI these days. The tools for predicting MS2 spectra are getting better. So even without having standards, that'll give you kind of a better look and save you some money to see what the MS2 fingerprint should look like, which should give you more confidence in the identified MS2 spectra.
Maybe one final question for you on this topic. Is there, we haven't heard anything about post-translational modifications. Is there a reason to think that we're missing those, or is there a modification that is commonly seen in those microproteins?
I mean, common is a tough word. None of these are commonly seen yet.
Anything that you see in larger proteins? Okay.
Yeah. So phosphorylation has been published, I think a couple times now, definitely by Sarah Slavov's group. I feel like, folks, maybe you can also remind me in case I've misspoken, but I feel like we've seen evidence of ubiquitination too, for at least some of these. But I have no reason to think that they can't be modified in the same way as larger proteins as long as they have the requisite sequences on them. And phosphorylation definitely should be there.
Wonderful. Thank you so much. And I posted, I think it did work now. I posted the papers for each panel member into the chat. So I think the one Thomas has mentioned is also now in the chat window. So Lee, I have a question for you next. What other organisms or tissues or cell types are people trying to perform RiboSeq on or have tried and potentially have failed? And what other methods could be used if RiboSeq is not amenable to your favorite organism to access microproteins?
Well, thank you very much for asking me this question, because while I was hearing Mara and Sebastian's great talks on ribosome profiling, I had very huge imposter syndrome. One of the reasons is in my lab, we actually have never successfully made ribosome profiling. If you check about the published papers, most of those papers were generated from yeast and mammalian samples. The general short idea is it's very difficult to do that from insects. The reason is for most of this kind of high quality ribosome profiling that we need to get this kind of periodicity, as Mara and Sebastian outlined, we need to use RNAs for this kind of digestion, but it doesn't work in Drosophila. So basically the problem is when you digest it, you don't normally get the monosome. It got degraded when the polysomes are not even fully digested. So a second way to deal with that is to use amylase. Amylase is workable in Drosophila, but the problem is because it has this kind of 3' AT bias, it doesn't produce this periodicity. And without the periodicity, it created the challenge that how do you make sure that this is not long non-coding RNA that just happen to be protected by ribosomes, or they are actually translated. So there is a very smart way by basically Juan Pablo Coso, and they basically use this kind of, they firstly enrich the polysome and then made them to monosome and then do sequencing. I think this is a great approach. The only problem is that if we can only isolate polysome, that will have a problem that if new proteins or short proteins, they're only protected by this monosome, that will be some challenge technically. So I think in my opinion, I think ribosome is very, very useful for identifying small proteins and some of the evolutionary young proteins. But depending on which clade you are studying, especially if you are studying in Drosophila, this is of just insects in general, I think there is a huge technical challenge that we are still trying to overcome. I agree with Sebastian's discussion and Thomas's discussion about mass spec. I think, for a long time, people still regard that as a golden standard. It's very difficult to deal with that, but I think the method is getting better and better. So maybe we can even revisit some of the old data that use new method to truly have a better way to analyze this type of mass spec spectra to make sure that we consider, but not over consider, problems related to post-translational regulation and other properties. So I think that's probably another directions that we are very interested in trying.
Great, thank you so much. So what I'm hearing is that the method is now very well developed for human or mammalian cells and tissues and systems, but it's still posing challenges for other systems. So your Drosophila work is fantastic in that respect. So please go and check that out. Great. My next question is for John Presner. And it could be like my ignorance of inviting people that mostly work on cancer, but there seems to be a bias in microproteins in cancer and immunology over other areas. We've seen some work on metabolism, and there's also fantastic work in mitochondrial function, but there seems to be a large role of microproteins in cancer. Is that because of the biology of the microproteins, or is it because of the people working in the field so far?
Yeah, thanks for that question. So I would agree. I think when you look at the biomedical sort of clinical aspect of this field, I see, I guess, several distinct groups being particularly highlighted. First, the cancer biology community, as you mentioned. Second, immunology, which you mentioned. Third, I'd add human genetics. And so there's a large portion of physician scientists and clinical researchers who are interested in unlocking either novel monogenic diseases through these or polygenic diseases by looking at variants in these. And is there sort of a biological bias of microproteins in these areas, or is this more of a sample bias of who the researchers are who are looking at them? And I'm going to say it's a little bit of both. So I think if you unpack this a little bit, I think the interest in the human genetics community is logical in that these communities are looking for novel mutations to explain patients who have not had mutations found in their canonical exomes. I think in the immunology and cancer fields, there's a little bit of biological bias as well, particularly perhaps in cancer, where we suspect, and there's a larger body of literature about what is ribosome dysfunction generally in a disease state, and how does that then influence what is being translated outside of the canonical genes, and even how does it influence translation of the canonical genes, things like read-throughs and stop codons and things like this. And so, I think in some of these disease sites, there is also a biological rationale as to why people might be particularly interested in them. But I think that there are also potentially some clinical scenarios where there are distinct patient populations that are being looked at as well.
Great. And maybe that might be more of a philosophical question, but the reason why microproteins seem to be enriched in certain cancer cells, is that because of the machinery going havoc, or is it the cancer inducing this change, or is it the cell making then potentially peptides that can be recognized by the immune system to specifically target those cells? Is there a consensus on that?
I think those are big open questions in the field, to be honest with you. And my honest sense is that any attempt to succinctly encapsulate those is going to be probably not accurate at this time, because I think that I think there's a lot of research going on in particularly those fields. One anecdote I can share in this regard is work in cancer and in transformed cells where they've been subjected to amino acid stresses, as what might happen in certain microenvironments within the tumor or other disease settings where you have poor oxygenation of the human tissue. You'll get amino acid stressors. And in that situation, there's an increase in misalignment of tRNAs with the ribosome, so that there's an increased ability to read through either stop codons or mistranslate certain codons that are under deprivation conditions. And so, those types of biological studies are actively ongoing. And I think in the next couple of years, we'll be able to answer your question maybe a little bit more solidly.
Wonderful. I think there's also a ribosome fall off under certain conditions, and that can also then lead to shorter proteins or microproteins in certain conditions. Great. Yeah, lots to do. Are there any questions from the audience right now? Otherwise, I can continue with some of the questions I have for the panel. I think there's one in the chat. Let me look at. Okay, I think there's one in the chat. Okay, we have a question from Alana. In the absence of proteomics data, is there a way to determine bioinformatically whether the differential up translation of sorts in a disease data set is due to ribosome stalling or an increased abundance in peptide production? Does anybody want to answer?
Well, I guess if you have ribosome stalling, then you'll see a differential pattern of the ribocycle reads. So you could in principle be able to differentiate. You should be able to differentiate between the two situations if you look for it specifically, of course.
So I guess I can add in some of the work that I think Sebastian and I have worked on collectively in our groups has looked at molecular subtyping of a certain pediatric brain tumor called medulloblastoma, where there was upregulation. And what we did observe in that case was that there was upregulation of translation of many open reading frames with certain molecular drivers of that disease. And so that might argue for uptranslation of the smorphs, at least in some of these disease settings. I think one interesting question that's kind of a follow-up to the one that's asked is then, does that upregulation of translation correlate with actually upregulation of the protein? Or are there other mechanisms on the back end of proteostasis that then also play a role in here? And I think that, again, is something that, continues to be sort of an open area for investigation.
I'd also ask if what looks like an upregulation of translation is also connected to, prior to that, an upregulation of transcription in the first place, depending on what the experiment is.
Great. I had my initial question to all of you was, will there be an explosion in microproteins once we have removed technical barriers? And just based on Lee's answer, it seems like there are still many organisms and tissues and cell types and conditions where RiboSeq has not been successful yet. So the answer would be yes, there will be an explosion. But in terms of the sequences that we already think, or let's look at the human system, are there surprises that we may encounter where there are lots of start codons we didn't really anticipate, or there's an intrinsic limitation of RiboSeq that doesn't capture all the ORFs that are translated? Are we expecting more peptides or microproteins to emerge in the future?
I think one interesting question is, how small can microproteins be? I mean, especially when I talked about the evolutionary arguments, if we talk about not just microproteins, but also potentially nanoproteins that are absolutely tiny, let's say under 15 amino acids, is there a whole world of these absolutely tiny proteins out there? If there is, how would we find them? Because it would be really hard to find with evolutionary arguments. And, the experts here will tell you that RiboSeq does have single codon resolution, so perhaps we can go in that direction.
I think Sebastian wants to say something. So that's John.
Yeah, thanks, John. I mean, I think Sonia Chotani is also in this call, and her work has shown that there are many of these really short ORFs. And I think in one of our recent studies, we also showed that there are these super small translation products, but they're so small that with mass spec you cannot detect them because they're just too small. But for some of them, we see that they're conserved all the way down to chickens, with the exact same coding sequence and also present in RiboSeq data. So that tells me that there is something that small that is present that is impossible to detect with mass spec, and that likely does something, and we have no idea what it is.
Yeah, I think that's one of the topics we didn't really touch on because we thought that it'll lead to too many discussions. But if you are interested, you should attend the upcoming conferences, the Keystone and the GRC. How do we ever get on top of understanding function, right? When there's all of a sudden this massive explosion of protein or peptide sequences, and most of these do not have any domains or any signatures that give away their function or potential function. So that's a whole different field that will hopefully advance in the future. John, you have one more comment? Where you have your hand raised?
Thank you. Actually, to dovetail on that, I just want to share, I guess, my one concern I have with the audience and trying to motivate maybe those in the audience to help take this up. I think with long non-coding RNAs and the invention of RNA sequencing, all of a sudden, within a couple of years, it went from a small number of long non-coding RNAs to 15,000 or something like this, thousands and thousands of them. And it took a very long time, and I'm not sure we're even there to know as a scientific community what to do with all of those, right? How do you look at them? How do you think about them? Do you just ignore them because they're overwhelmingly too many of them? And I worry about the same thing with microproteins, that there might be an overwhelming number of them. And so one, I think, task to the research community, to those of you who are on this call, is to think in your own life, in your hands, in your research, how can you think about ways to grapple with these, to bring them across the bridge from sort of mere computational detections to something that we should actually care about and convince us as a scientific community. Because it's hard work, and I think that's work that's very important to do.
Right. Thank you, John. Billy, I think we have time for one more comment.
All right, I would just have a very quick comment because I really like Jonathan's comment about if proteins are very small, how do we consider them evolutionarily? Because there is a practical challenge that, if the proteins are small, they don't have to be nanoproteins, even they are just small enough. Sometimes this kind of classic evolutionary or comparative genomic tools or molecular genetic tools they truly couldn't distinguish if a gene is evolving neutrally or under purifying selection. You know, as Mara very nicely outlined, we wanted to find those ones that are functional, but sometimes there might be a difficulty in the future that some of them, they don't really seem to be evolving under purifying selection because they are small. It's hard to do statistical analysis, but they are in fact quite important.
Right, yeah. I remember that from the conferences too, that the positive selection is not always measurable, but yet some genes are essential in yeast, so that's kind of contradictory. Great, I think we have some, the links to the upcoming conferences in the chat window, so people please go, and if you're interested in the field, read the papers that we posted. Thank you so much for the two speakers today and the panel members for this insightful discussion, and for everybody who attended. I think I'll give the Zoom room back to Lauren, who has a few closing remarks. Thanks again, everybody.
So thanks everyone
Becky.
for, yeah, it's okay. Thanks everyone for coming today. I'd like to let everyone else know that we do have another virtual seminar that's coming up in May 22nd. It's on immunogenicity of peptides and peptide impurity. And Brian Roberts from Epivax will talk, as well as Daniela Verthehi from the FDA, to just talk about immunogenicity and peptides. So we're really looking forward to that. So look at on the BPS website, or stay posted for when that's coming up. Okay, thank you everyone for a great talk.